I'm Cathy Clabby, a contributing editor at American Scientist, the magazine of Sigma Xi, the Scientific Research Society. Sigma Xi awarded the 2012 Walston Chubb Award for Innovation to Joseph DeSimone. A distinguished and entrepreneurial chemist, DeSimone holds faculty positions at the University of North Carolina at Chapel Hill and at North Carolina State University. He is director of the Keenan Institute of Private Enterprise at UNC. Professor DeSimone has a long history of innovation, most recently in the life sciences. His early experiments with liquid and supercritical carbon dioxide led to a greener dry cleaning method and a less polluting means to make a Teflon-like material. Today, his research team focuses on new approaches to making medical devices, vaccines, and medicine. This work starts on the nanoscale and uses fabrication processes pioneered in the computer industry. The approach holds promise in vaccines, cancer treatment, inhalable medical treatments, synthetic blood, and other medical products. DeSimone has commercialized several inventions and has helped start multiple companies. The Bill and Melinda Gates Foundation has invested $10 million in one company, Liquidia Technologies, for vaccine development. Professor DeSimone has won numerous awards, including the Lemelson MIT Prize, which recognizes people who turn good ideas into tangible products that help humanity.
My high school in Pennsylvania, we had scholarships from a local liberal arts, a residential liberal arts college in our neighborhood. Ursinus College had some scholarships available for townies. And got a partial scholarship to Ursinus College. Didn't live there; we couldn't afford it. I still worked, and parents worked, and lived at home and was a commuter. But fell in love with chemistry there, and great profs, and got exposed to the liberal arts education there, and that was the beginning of it for me. It was a narrow idea that grew, and again, it's having your head up, looking about you, and seeing connections that may not be there when you first thought of something.
Yes.
But the environmental pressures in the early '90s on the chemical industry were really ramping up. I also recognized that DuPont in particular was getting battered by the manufacturing process for making Teflon. And this process turned out to be really good for that class of polymers, that class of plastics, fluorinated materials. And so we made a connection, the intellectual connection, that this could be an environmentally friendly way to make that material. That was a technologically important material. And so that became the genesis of Key Paper in Science in '92. And then I wanted to make more plastics in CO2, not only for the environmental reasons, but for a utilitarian reason. We thought it was going to be better materials, better way of processing. We wanted to make paint in CO2 instead of water, so that we could dry faster, spraying out of CO2, instantly dry. CO2 is thought of as being, in a solid form, as dry ice, right?
Yes.
So having CO2 as a dry liquid would allow us to use chemistries that are unstable in water, and so that we could get into some important new coatings. And in order to do that, we needed surfactants or soap molecules for CO2. But we did that for making these paints in CO2. And we had a paper in Science again in '94 and '96. But then the soap molecules for CO2 in and of themselves was new. We needed it for making the paint in CO2. But we also knew then that people were thinking about using CO2 in cleaning beyond the coffee extractions.
Yes.
But like water is good for is a good solvent for certain things, but you need soap. CO2 is going to be good for certain cleaning, but you also going to need soap too. So we decided to take that soap, use it for CO2 and cleaning. and you know, we had partnered with DuPont in making the Teflon. And my students and I in the early days were thinking, well, we could do cleaning. People are interested in cleaning. Can we just license it to them? Well, you know, there's no DuPont or IBM of the cleaning world, dry cleaning in particular. People were trying to clean clothes in CO2 and needed soap. But there was no one really to license it to. And we needed to work in certain conditions. And so we decided to launch a company back in 95, 96 to start doing CO2 and cleaning. And that launched MyCell Technologies. And, you know, we raised a lot of money. We built, you know, we had to invent dry cleaning machines then, too. And then you sort of realize all the things you have to invent. You can't just put CO2 in a normal dry cleaning machine, right? It needs to be a pressurized vessel. And so we were inventing a lot of stuff to make this whole thing work. And to make a long story short, we had a terrific dry cleaning machine. We built 70 of them. We were out of the gates. This was the, you know, the dot-com go-go days, and but we had something tangible, and you know, we were going to revolutionize the dry cleaning sector. And so we were doing that, and then I had a colleague in the medical school asked us about could we do some things with drug delivery and something called gene therapy. And you know, so I said, yeah, I'd love to do something else. I was interested in biology at this point, and I had a student, and we started looking at ways of making particles for gene therapy. And it was ironic that everybody up to this point was using these emulsion techniques, which to me in my world is a cousin to paint. We're going to deliver nucleic acids for gene therapy, and the best way to do that in the literature was something to me that looked like a cousin to paint. And there was such an intellectual gulf between this high-tech gene therapy technology and paint that there had to be a better way. And we published that paper using the old technology, but you know, now we start molding particles. Couldn't we bridge the two? And that's what, you know, we had a paper in 2005 that married this idea of molding particles for drug delivery. And we were filing patents in this new area. So there was a connection from the CO2 world.
Yes.
There was a connection from the stent world and medicine.
Yes. An
overture from the medical school. Everything coming together, convergence, and allowed us to move in this whole new area. Yeah, we didn't know it at the time, right? Again, we're filing patents. I thought it was going to be important. And on a micro scale, this liquid Teflon was being used to make something called a lab on a chip. And we thought that was going to be a big part of Liquidia, more material science. Come to find out that lab on a chip technology and industry really hasn't emerged very large. In fact, the one product it's going to be in is going to be on a device for NASA on the search for life on Mars in a 2013 mission. That's not a big market.
There won't be many of those missions. Yes,
they need a couple. But you know, offshoots of that started going in more and more towards, you know, therapeutics, and that's, you know, it's really blossomed. And so you know, my whole lab transitioned to life sciences, and we had good funding in the CO2 world, and we shut it down, which is, you know, risky. And I was able to successfully get funding from NIH and the NCI and National Cancer Institute, National Institutes of Health. You know, we started getting some of our particles. We started shipping them over to the medical school to a physician collaborator. And he said, you know, we got together, and he said, well, it didn't work. Well, what happened? Well, it just didn't work. That's not going to be very helpful to us. So realized at that point that if we're going to do something here, we need to jump in ourselves and learn biology. And so, you know, again, a long story short, we have all our own cell culture facilities here, and my students are trained in animal core facilities, and I have MD, PhD students, and Most of the lab is pharmacy students and a lot of chemistry students and chemical engineering students, and so we are well versed in biology now and are still learning a lot. But it took that commitment to learn these new areas in order for us to actually move the ball down the field. Because if you just can't hand stuff off to somebody else and get it didn't work, you got to do it yourself to understand why it didn't work, what you were going to be able to change to make it work. And what you now find out in areas like vaccines, that there's a lot of immunologists and vaccinologists that work in this field, and nano and microparticles are actually an important part of vaccine design. But they often get particles because that's what happens. They're not done with a clean sheet of paper. You know, if you ask a vaccinologist, immunologist to, okay, with a clean sheet of paper, actually design exactly what you think would be the best particle. The right size, the right shape, the right chemical composition. They never did that exercise. They had aspirations of what that could be, but they could never get that particle. Well, we can make that with our templated approach. So we can actually take these sort of blue sky, we'd like it to do this. Well, we can give you that. And now we actually can design particles that are inspired by biological processes to do those functions. And it's turned out to be a fantastic strategy. You know, it's actually both. You know, doing it yourself is a big part of that, but also partnering with domain experts, bringing people into your team that are black belts in these other areas, and so, but committing to learn what they know and vice versa. And your partners and collaborators also have to be reciprocate that quest for or thirst for knowledge in your domain too. So that's a two-way street. And once you find those people, you can sort of hang on to them, and you become, you know, sort of Batman and Robin, and you can sort of do your thing. And Tom and Jerry, whatever the analogy is, but yeah. So we look for those kinds of collaborators, and that's key.
Yes,
that's really key. But you know, I think too, the translation of one's science into the private sector or commercial sector or venture-backed companies is a big step, and one that, you know, it's not for everybody because it's hard. But in academia, you know, we publish papers, and you know, it's peer review, and peer review is really rigorous. But once you venture out into starting companies, I argue that's peer review on steroids. Because the level of scrutiny one gets when people are betting sometimes their own money on your ideas is really extraordinary scrutiny. And the best venture firms will line up, you know, Nobel laureates and other people to drill you on, you know, why are you doing it that way? Where are you going? I mean, asking really hard questions, and it elevates your science. So I find that, you know, spinning out companies makes us better scientists, allows us to see things that many academics don't get a chance to see, and understand where the market needs are in a crisp way. Understand the ability to articulate the value proposition very succinctly and clearly helps us on grant writing. So, you know, I think it's really synergistic with my academic lab that we're so active entrepreneurially, because it makes us better scientists. And the other skill, back to the earlier discussion then, is, well, you know, recognizing that vision without resources is a hallucination. Right? So if you have, you know, if you believe that you have all these opportunities, the other skill then you bring to this group is, how do we finance it? Understanding the economic models that will allow us to effectively engage in that and make that vision happen. But early in my career, I was invited to give a lecture, to participate in an innovation team. Innovation is not a new word, right? So this is back in early 90s at a large chemical company in Europe. And I went to participate in this team, and it was a pretty sterile-like environment. It was a boardroom, big oval table, and I'll never forget it. It was a bunch of white guys that graduated all from the same 2 research groups in Europe. They knew exactly the same stuff. And I thought, boy, this was their innovation group at a large chemical company. I thought, they're at a structural disadvantage to being innovative if everyone around the table knows the same stuff. I saw them resonating with each other in this dialogue, and I was talking about something very different than what they knew, and I was almost dismissed. I felt like I flew in from Mars, right? And I thought, boy, that's not a very open environment. So it started crystallizing for me early on that, we learn the most from those that we have least in common with. If you're trying to maximize learning, and that in fact diversity is a fundamental tenet of innovation. It's key. And whether it's different expertise, different cultural backgrounds. I mean, I have students that are mechanically terrific with their hands, and whether I'm interested in those that have changed car tires and changed oil, worked on their cars. That's a different type of hands-on student. And I've got other students that are more theoretical and more analytical, but getting that group to work together. I have students that come with a much more deprived country maybe, and socially economically challenged environment. They think about problems differently than somebody who's grown up with a lot of money. They just do. And to recognize those differences and to embrace them as a strategy to come up with better solutions is what we try to do. But I had a chance I knew I was going to meet with Bill Gates, or I had a chance to meet with Bill Gates at a meeting.
When was this?
This would have been about 2.5 years ago. And I worked real hard to get myself into that meeting that Bill was going to be at. And then I knew I was going to be introduced to Bill. I knew this for 3 months ahead of time. And so, just in the back of my mind, what would I say to Bill Gates to capture his interest and attention? So, and you had 10 seconds, right? So this is the ultimate elevator speech.
A short one.
Short one, right? So, Bill, this is Joe. Joe, this is Bill. I know who it is, and Bill said, what do you do? And I said, Bill, we're using the manufacturing techniques of the computer industry to make better vaccines. So he obviously knows something about computers. And the Bill and Melinda Gates Foundation is all about vaccines and global health. And so this was a direct shot at both of them. Now he's fueling the global health initiative with the financial aspects of Microsoft, but this was a tangible technical connection of using the manufacturing techniques of transistors and computer chips to make better vaccines. And that's this molding technology. We call it's lithography. It's an offshoot of the computer industry. So we're trying to take the techniques from making computers and apply them to make vaccines. Well, that got his attention. Turned into almost a 30-minute conversation.
How are we doing?
Yeah, I don't buy that. I think we're being. This is a great place to be innovative, and I think at its core, we're still doing the right things. Where we're slowing down is the translation of that. There are some real important issues out there that are making this swimming upstream here. You look at the FDA and the regulatory issues. That stent I mentioned to you, that bioabsorbable stent.
Yes.
that was brought forth in Europe and Australia and New Zealand first. Got CE mark approval, which is the FDA equivalent in Europe, for sale. And when all that body of information was presented to the U.S. FDA, Abbott has got 70 plus questions back. It's going to delay it for another year and a half. It's already approved in Europe. It's going to cost, it's a multi-billion-dollar product that the American people are not going to have access to. It's the biggest breakthrough in the stent marketplace. And so, that's a big issue. And so that's a problem. But I'll even go further. We have a new device that I'm really excited about, that we have, with the stents I learned about catheters and deployment. I'm doing oncology now, 2 pieces. We had a partnership with a company in Massachusetts that used a mild electric current to drive drugs deep into the eye for wet macular degeneration. It's called ionophoresis. And it's painless. Put a device on your eyeball, out to a battery pack, counter electrode in your forehead, hold for 10 minutes, drug goes deep into the eye. 3 disparate things, right? Catheters and stents, oncology, ionophoresis of the eye. Come to find out, one of the cancers that we really want to treat is pancreatic cancer. These tumors don't have a blood supply. The bloodstream is the highway to get drugs in and nanoparticles in. They don't have a blood supply. Those tumors are as white as your eyeball, interestingly. So we came up with a device that combines catheters, electric field, chemotherapy agents, and it's surgically implanted, working with my colleague Jen Ye in the medical school, who's a surgeon. And we can surgically implant that device on human patient-derived pancreatic cancer tumors, surgically implanted and growing in mice. And no drug, no chemotherapy that they have works on that tumor. Use our device, we're now seeing 80+% tumor regression in pancreatic cancer. Very exciting. We want to start a company called Interventional Oncology. A lot of the big pharma companies that want to finance this company want us to domicile it in Europe or somewhere else, because they can use their overseas profits to finance the company. If they finance it in the U.S., those dollars, when they get repatriated, have a huge tax rate. So one thing is our technology is being commercialized elsewhere, but now they want the companies to be domiciled elsewhere. I still think we're great on the innovation process.
Yes.
But whether the economy is going to benefit from that is the story, is the negative story. Because not only if our companies are going to be located, not only if the technologies have to start there, if now the companies have to start there.
Yes.
Those companies then, as they develop manufacturing and begin to scale, they're not going to come back. And there's going to be a lot of innovation at those companies while they manufacture. And one thing people, they say, well, outsource manufacturing. Well, when you manufacture, you innovate.
Yes.
And if you're not manufacturing, you don't have the opportunity to innovate in your country. And so it can be a self-fulfilling issue here. And that's so the innovation process, I think, is still strong.
Yes.
But now we've got these other issues that are going to get in the way of the economy.
These other barriers.
These other barriers.
To helping these good ideas develop, blossom here.
Yeah. So a lot of people are staring at this. Jay Walker, who does TED Med, and I spoke in TED, and others are really driving these issues. And Senator Kay Hagan had a bill for a tax holiday to allow companies to, as a tax holiday, to bring those dollars back. If you had a tax holiday, there'd be this massive investment of overseas dollars here in the United States. It wouldn't cost anybody anything. It would really goose the economy and drive these innovations forward to drive the economy. Well, we got to get over these issues. Otherwise, our innovations here in the United States will, in fact, be building the economies elsewhere.
Well, we at Sigma Xi wish you the best in all of that, and thank you so much for taking the time to talk to us, and congratulations for winning this award.